Mi nombre es Ángela Sastre, soy catedrática de Química Orgánica de la Universidad Miguel Hernández de Elche y directora del Instituto de Bioingeniería de la misma universidad. Y bueno, pues es un placer para mí estar hoy aquí en estas jornadas. Y bueno, lo primero de todo, me gustaría felicitar al ICMol por esos números presentados y por ese número 1 en el ranking, no solo a su director, sino a todos los miembros y investigadores e investigadoras del centro. Creo que estáis haciendo una labor encomiable y sois un ejemplo a seguir, ¿no? De hecho, yo siempre os pongo de referencia y es ahí a donde quiero llegar, por supuesto. Bueno, se me ha encomendado la labor de presentar al primer conferenciante de esta jornada, de esta estupenda jornada, y es el profesor José Luis Mascareñas. Aquí tenemos, vamos un poquito tarde, pero bueno, no pasa nada. Se graduó en Química en el año 84, y también en la Universidad de Santiago de Compostela, y recibió su doctorado en la misma universidad en el 1998. Realizó una estancia postdoctoral en la Universidad de Stanford, en Estados Unidos, bajo la supervisión del profesor Paul Bender en el 89-90. Después se incorporó a la Universidad de Santiago de Compostela como profesor titular de universidad en el 93 y unos años más tarde, en el 2005, recibió ya, bueno, pues, consiguió el full professor, la cátedra en Química Orgánica. Y es director del Centro de Investigación de Biología Química y Materiales Moleculares del CIQUS. También ha realizado distintas estancias en la Universidad de Harvard de investigación en el verano, en el 92 y 95, y también en la Universidad de Cambridge en el 2009 y en el MIT, en el Massachusetts Institute of Technology, en el 2013. En cuanto a sus números, pues ha publicado más de 170 artículos y ha dado más de 120 conferencias invitadas, pero creo que lo que más le gusta es la supervisión de doctorandos, y 18 postdoc, 10 están en el grupo, 31 tesis, 11 están en marcha, más de 55 estudiantes de máster y estudiantes de grado, y bueno, pues 5 de sus estudiantes han conseguido la Ramón y Cajal y 3 pues han sido premiados por la Real Sociedad Española de Química como investigadores jóvenes. Es miembro de distintas sociedades académicas y bueno, del grupo de gobierno y demás, y también es fundador presidente del Grupo de Química Biológica de la Real Sociedad Española de Química. En cuanto a sus premios y distinciones, pues en 2019 recibió el Premio de Química Orgánica de la Real Sociedad Española de Química. En 2013 recibió la medalla de oro de la Universidad de Santiago de Compostela. En 2013 también, en ese mismo año, recibió un RSI Advanced Grant con su proyecto MetBioCat. En 2014 recibió, fue nominado como Gallego del Año por el Grupo Correo Gallego, y pues en 2015 recibió la medalla de oro de la Real Sociedad Española de Química, que es uno de los reconocimientos más altos en química a nivel español. Y también ese mismo año el premio de Félix Serratosa Lectureship. Bueno, su investigación se centra en síntesis orgánica para el descubrimiento de nuevos sistemas basados en catálisis metálica y también química biológica, enfocados al desarrollo de estrategias sintéticas para el estudio de propiedades biológicas. Así que, bueno, pues por último, pero no por ello menos importante, hay 3 cosas que creo que compartimos. Primero, que tiene un amor muy fuerte por su pueblo, yo también. Él nació en el 61 en Allariz, en Orense, y también yo nací en Segovia, en un pueblo pequeñito, un poquito más tarde. Pero bueno, no mucho más. Y bueno, pues también su amor por el baloncesto, me ha dicho que es gran seguidor del Obradoiro Club. Y bueno, pues también mis hijos juegan al baloncesto y en el club ilicitano. Y sobre todo, lo más importante, creo, es su amor por el CIQUS. Creo que es algo que es algo bueno, pues que tiene bueno, pues él, su director, y bueno, pues se fue creado en el 2010, y bueno, pues también es parte también de que la Universidad de Santiago de Compostela tenga ese reconocimiento de Campus Vida. Entonces él, pues, bueno, pues muchas de sus energías van enfocadas a eso. Entonces yo pues también soy directora de instituto y también pues tengo ese placer de poder bueno, pues trabajar en la investigación. Así que nada más, cuando quieras, el floor is yours y muchas gracias. Y bueno, pues nada más.
Buenos días. Voy a hacer los agradecimientos en castellano y después la charla la voy a hacer en inglés. Primero, muchas gracias, Ángela, por la presentación tan detallada. Efectivamente, me gusta el baloncesto. Tengo mucho interés en convertir en CIQUS en un centro de referencia y estoy muy orgulloso de mi pueblo. Está bien. Los números, yo los he visto por ahí, están un poco viejos. No tiene mucha importancia, pero por ejemplo, en lo que sí tengo mucho orgullo, que es en la formación de gente, ya llevo 9 Ramones y Cajales y 2 ERC Starting Grant, que eso sí me produce mucha satisfacción porque justamente es una de nuestras grandes labores y 40 tesis doctorales. O sea que ya me he hecho más viejo. Lo segundo, pues nada, solo tengo palabras de agradecimiento a Eugenio y a Enrique, no sé dónde está, ahí, por la invitación. Fue todo un poco apurado, me cogiste así casi por los pelos, pero estoy encantadísimo de estar aquí. No puedo más que darte la enhorabuena por lo que estáis consiguiendo con el ICMol. Después hablamos del Nature Index, pero realmente es impresionante ver el progreso y sobre todo que es esa fuerza de la gente joven con toda su creatividad y que le demos el campo de juego para que puedan realmente demostrar todo lo que valen, creo que es lo más importante. O sea que enhorabuena. Bueno, a ver si salimos todos indemnes de la Junta después teniendo en cuenta el acoso del virus. Espero que sí, porque estáis tomando todas las medidas adecuadas para que esto pueda ser así. Okay, I'm going to switch to English to start my lecture. It is difficult to give a dissemination lecture and also at the same time speaking about your research, but I will try. I'm also considering that my research is not in the same focus that the main areas of ICMol, but I will try. Okay. So okay, let me start by recalling a famous book written by the also famous scientist Esbjørn Rødinger in 1944, entitled "What is Life?" So this book was after a series of lectures of Rødinger in Dublin. He was exiled by the Nazis, and he was quite controversial because people like the famous Linus Pauling were criticizing the book because he said, "Oh my God, what is a physicist writing about something that belongs to the biology field?" You know. However, for other people like Watson and Crick, that 10 years later discovered the structure of DNA, the book was very inspiring because in the book, Rødinger starts to propose some kind of primitive ideas about the genetic code, and this was very useful for Watson and Crick. Anyway, you could ask, did he answer the question? As you might imagine, he didn't answer the question. I mean, it's very difficult to answer what is life. But the problem is that there has been almost 80 years, and we are still struggling with the same questions. What is life? How did life emerge in Earth? Of course, how to make life. We really cannot define what is life. This is like a famous judge in the US in the 60s used to say about pornography, that I cannot define it, but I know when I see it. The same happens with life. With life. We can recognize if something is alive. So it's difficult to define, but what we can do is try to identify some characteristics of living things. With all the observations, I'm going to name a few. Living organisms are able to replicate and reproduce, at least as we know. Then in Earth, I'm not sure what will happen in Mars, but there is a cellular organization in living organisms. They respond to the changes in the environment thanks to the homeostasis, and of course, they follow the laws of evolution and natural selection. But there is a very important characteristic of living things. That is this one, metabolism. What is metabolism? Metabolism is the network of chemical reactions that provide the food and the energy for cells to live. There is a very interesting discussion in the origin of life right now on whether it was first the metabolism, I mean the reactions, or it was first the information, I mean the DNA or the RNA. It's not clear. There is a discussion on that, and it's very interesting. If you like the topic, you can go to the internet and see the different views. Anyway, the metabolism is very important. As you might imagine, the cellular metabolism is very complex. I could draw something like this. This would be a cell. This is a typical metabolism of glucose that is converted to pyruvate, and then pyruvate goes to the mitochondria. And I can take this out, no, I think. And in the mitochondria, they is converted to energy, to ATP. So you might think, well, this is very complex. But I can tell you, it's not as complex as this. It's much more complex. If we really want to represent how is the metabolism of a cell, we have to go to a drawing like this. So it's a very complex network of chemical reactions. Each dot will be a metabolite, a biomolecule that is converted to other through a chemical reaction. And these chemical reactions can take place spontaneously, in our case at 77 degrees, thanks to the catalyst of nature, thanks to enzymes. And there are over 1/3 of enzymes that contain in the active site a metal. And these enzymes are called metalloenzymes. And I can show you here one of these enzymes, of these metalloenzymes. This is my favorite. I'll call the hydrogenase. Probably I'm here right now thanks to him, thanks to this enzyme, because it's in the liver and it's taking care of all the ethanol that we had yesterday in the dinner that is converting the ethanol into less toxic acetate. So it's a very important enzyme for us. No. So as you can see, this enzyme contains zinc in the active site, and the zinc plays a key role because the zinc in this case is behaving as a Lewis acid, is binding to the alcohol, and then after the interaction with the zinc, the oxidation to the aldehyde is much better, much easier, and the aldehyde is further oxidized to acetate. So this is a metalloenzyme, a natural metalloenzyme, and if you go to the periodic table, we can see other metals that nature uses to build the other metalloenzymes. And most of the metals used by nature are zinc, copper, cobalt, iron, in some cases manganese and nickel also. So, and these metals behave like Lewis acid or as redox centers. But if you look at the periodic table, there are many other metals, but nature decided to use just a few. This is another interesting discussion. We can discuss about that. This is why only these ones. But I want to call you the attention especially about these metals here, of the second and the third row of the periodic table. Why these metals? Because these metals are extremely powerful for promoting organic transformations. These are the metals of organometallic catalysis, the more powerful metals to perform reactions that are unknown to nature, organometallic catalysis. And why are so relevant these metals? Because they have specific electronic properties, coordination properties, redox properties that favor many types of mechanisms that do not exist in natural enzymes. However, the main mechanism for this metal, for these catalysts based on these metals, is the same than enzymes. So the metal, the transition metal of the metal complex, binds the substrates in the active center in the coordination sphere, and when the substrates are in the coordination sphere, they are activated. And once they are activated, they react, they form the product, and the product is released to the medium, and the metal catalyst is regenerated. So this is the mechanism of the transition metal catalysis. It's similar to that of the enzymes. So, coming back to the metals, I think these are very important metals for humans, for promoting human-invented transformation, not nature-invented transformations. And to reinforce this idea, I'm going to show you some examples of practical applications of these metals. Maybe you didn't realize before, but for instance, thanks to these metals, we can use the catalytic converters in cars, because these metals are able to convert the toxic gases that come from the combustion to less toxic gases, even though CO2 is not the best one, but it's better CO2 than CO, you know. So, and this is because platinum, palladium, whatever, are doing redox type of transformations. But however, one of the fields in which this type of metal catalysts have found more applications is in the pharmaceutical industry. And I'm going to show you a couple of examples. For instance, this molecule here that is called tazarotene is used for the treatment of acne or psoriasis. And we can have this drug in great part thanks to an organometallic catalytic reaction. In this case, a palladium catalyzed coupling between this type of precursor is an alkyne and a chloropyridine, and we are making a carbon-carbon bond. If we don't use this reaction, the number of steps to make this product is much longer, so it will be much more expensive. So we are taking advantage. This is a great application of a metal catalyzed transformation for our daily life. This is another example. This drug here is used every day in the hospitals for the treatment of specific type of leukemias. And we can make large amounts of these compounds, again, thanks to a metal catalyzed transformation. In this case, again, a palladium catalyzed coupling between this amino group and a bromo arene. So we are making this carbon-nitrogen coupling thanks to a palladium catalyzed process. So I could say many more processes that are very relevant for our daily life that are promoted by metal catalysts. But if you allow me, I'm going to show you more examples from my own research group, just briefly. I'm sure that most of you that studied basic organic chemistry know very well this transformation, no? The Diels-Alder cycle addition. No? Everybody knows the Diels-Alder cycle addition. It's a fantastic transformation. Diels-Alder won the Nobel Prize for that. It's fantastic because you just heat this precursor, and just by simple heating, you obtain this bicycle containing 2 new carbon-carbon bonds and 1 cycle. We increase the skeletal complexity of the system just by simple heating. But the Diels-Alder transformation has a limitation, has limitations. What is the limitation? I'm sure you know also that if you don't have this electron withdrawing group here, then the LUMO of this dianophile is higher, and then the reaction doesn't take place. So we need an electron withdrawing group in this position. So it's limited to specific substrates. How can you solve that? We thought that one way to solve that is changing the dianophile from an alkene to an alene, because this is more strained. However, when we did the reaction in the lab, if you heat this, you get mixtures. You get the starting material and mixtures of process. And now is when the magic of metal catalysts come into play. And we found that if you treat the same substrate with a small amount of a gold catalyst or platinum dichloride, in this case you need to heat, in a single step, you get this other type of cycloaddition. This is a different one than the Diels-Alder, because if you pay attention, we are not making a 6 but a 7-member ring. The 3 carbons of the allene 1, 2, 3, 1, 2, 3 are now in the final 7-member ring. And this is possible thanks to these catalytic amounts of gold. Why? What is the gold doing here? Well, the gold is activating. So when this allene is in the presence of gold, it gets nervous, it's activated. And by activation, you are able to make this intermediate that is a gold-linked allyl cation. And when you have the gold-linked allyl cation, it can undergo the cycloaddition reaction to give the 7-member ring. And now these are normal steps in organometallic chemistry, elimination, 1,2 hydrogen migration and elimination to yield the product. So this is a fantastic transformation, because with just the magic amount of gold, you build a very complex cycle. And this allows us to shorten the synthesis of a quite interesting natural product, that is this product here that is called Engerin A. It's a product that is right now in clinical phases for the treatment of cancer, renal cancer. And we were able to make a total synthesis of this compound thanks to our catalytic reaction. This is the catalytic reaction that I just showed you. So because of this catalytic reaction, we are able to build the basic skeleton of the molecule, and this shorten the asynthetic axis. Our synthesis has several advantages over others that were developed before. For instance, one of the advantages is that it's shorter, is more efficient, and also is very versatile, because if you change here conditions in the last step of the synthesis, you can make many analogs of the products. And this is important because some of these analogs present even better biological activities as anti-cancer compound than the natural compound. Indeed, we are right now making assays with this one here that is significantly more active than the natural compound. But with organometallic catalysis, you can not only activate double bonds, unsaturated bonds, pi electrons, you can also activate, and this is quite impressive, inert bonds. For instance, this is an inert bond, a carbon-hydrogen bond. We can activate this and cleave this carbon-hydrogen bond and make this type of metallocycles. And these are very important intermediates because they can be trapped by external alkenes to give, again, 7-member rings. And we made different type of applications of this conceptual idea, and I'm going to show you just one. This is a one-step synthesis of benzodiazepines that are very important pharmacophores in a single step, just by mixing this allene with this aniline and a palladium catalyst, we can make a great variety of benzodiazepines. But one of the wonderful things about organometallic catalysis is that by changing the metal, using other metal than palladium, or by changing the ligand, you can completely change the reactivity. And for instance, you use the same substrate, but instead of palladium, we use rhodium, and we use this specific ligand that we designed because it's an electron-deficient ligand. The product is different. It's an indolene. It's not the benzodiazepine that we made before. And this is because we changed the mechanism. The CH activation step is the first step. We cleave the CH bond, we make the metallocycle, and now we insert the alkyne. And now, because of the presence of rhodium and this ligand instead of palladium, the mechanism here is different. There is a migration of this hydrogen from this position to this position to give the product. And we can go further in this type of chemistry. Half of my group is working in this area. This is just the last example that I'm showing you, is that we can even achieve asymmetric catalysis by using chiral ligands in the metal. In this case, with palladium, you use chiral ligands. You can make this type of products that are isoquinolines with optical activity. I mean, you have 2 enantiomers of the product, and you only make one of the enantiomers of the product. This is something that every time every day is more important because many drugs are in the 2 enantiomeric forms, and only one of them is really the active form. Okay, this just was a brief summary of some of the things that we're doing in organometallic catalysis, but the main message that I want to drive to you is that this is a very powerful tool, okay? So how do we make the reactions? We make the reactions in round bottom flasks, in organic solvents, usually under air, and, under argon. I mean, we don't want to have oxygen, and also we don't want to have water. We want to avoid that. This is typical of organometallic catalysis. You don't want to have water, you don't want to have air, because the organometallic intermediates can be decomposed. But over 10 years ago, I was discussing with one of my students, I said, would it be possible to transfer, to translate, to export some of these reactions promoted by the second and third row or metal catalysts to the same environment in which metalloenzymes work, for instance, to a living cell? The student said, no way, it's impossible. Why? Because a living cell is a very complicated reaction medium. Look at a living cell. How is a living cell? We have the membrane, we have the nucleus, we have many things here. It's aqueous, it's crowded. Can you imagine how crowded is one of our cells? It's full of things. It's really not a container, it's a gel full of things. Maybe you are not lucky and you use your substrate and you add your metal, and maybe your substrate goes to the mitochondria and your metal goes to the nucleus, and then they don't meet. So this is the reason why the student didn't want to work in this topic. But you know, there were some precedents that some metals were compatible with water. Later on, we discovered a paper published by Eric Megers in Angewandte 2006, demonstrating that by using a Reuterium catalyst, he was able to remove this protecting group from a rhodamine to give the free rhodamine. This is not fluorescent and this is fluorescent. But the more important aspect of this paper that Megers publishes is that they were able to do this transformation in the presence of cells, of living cells. Even though it was not clear in that paper if the reaction was taking place outside or inside the cell or in the membrane, it was not clear. But for us, it was a very good reference because it was an indication that at least we could have some compatibility. No? And we started to work over 2010, 11 in this area, and just in 2011, there was a paper, it was the first paper really applying a palladium catalyst, even though this is a palladium nanoparticle, to promote the same transformations, and in this case, the authors were claiming that the reaction was taking place inside the cell. We were already working in this area, starting to work in this area. As part of our work, we had another research line in the group in the area of DNA binding. We were trying to develop new type of DNA binders based on peptides or small molecules, and for instance, is DAPI. This is a well-known DNA binding molecule. This is used in many molecular biology laboratories. Why? Because this molecule interacts to DNA, as you can see here. It inserts like cheese in a sandwich in the minor groove of the DNA. But the nice characteristic of DAPI is that by insertion into the minor groove, it is more fluorescent, and therefore DAPI is very often used to stain the nucleus of the cell. So this is the typical staining of the nucleus. I'm not sure if it's possible to go down in the light, because I'm not sure you can see the color. You can see it? Yeah. Okay. So this interaction with the DNA, for this interaction, it is critical the presence of this amidinium that is positively charged and make hydrogen bonding and electrostatic contacts with the edge of the bases. Then Matteo, there was a student that was talking to you about, he got the ERC starting ground now. Matteo was making this derivative in which he protected the amidinium with an analog protecting group. This is analog, so we remove the charge. And the curious thing is that now this compound
this compound, when you treat cells with this compound is not in the nucleus, this is the nucleus of the cell, but it's in the cytosol. So by introducing this moiety, we completely change the localization properties of the molecule. As you might imagine, our next step was adding the Reuterium catalyst to see whether we could recover the DNA binding. So we added the Reuterium catalyst, and we are happy to see that by adding the Reuterium catalyst, now we recover the DAPI, and we recover the staining of the cell nucleus. So for us, this result was quite exciting because it was one of the first demonstrations that we could use a small catalyst to carry out reactions inside living mammalian cells. We were struggling to publish this paper for more than 1 year, and finally, because the referees were always asking for new things, but finally we published it in 2014. So with this result, we opened a new field here, and many projects came to our mind. We had different ideas. For instance, one of the questions that we asked is, can we catalyze a reaction in a specific cellular organelle, not in the cell, but in an organelle? And we consider mitochondria. Why mitochondria? Because mitochondria is the energetic central of the cell. It's where ATP is made. So it's a very important cellular organelle. Can we promote a metal catalyzed reaction in the mitochondria of living cells? Well, it seemed difficult, but we try. And for that, my postdoctoral students in that topic made this compound. You don't need to understand, but it contains 3 parts. This is the Reuterium, this is the catalytic part. The Reuterium is a second generation Reuterium. This red part here is a mitochondria targeting unit. So this type of phosphonium with hydrophobicity usually accumulate in mitochondria of cells. And this is just a fluorophore that allows to track, to monitor the presence of the compound in the cell. Indeed, if you add this compound to the cell, you see the blue color, and you could say, well, this is mitochondria. Well, to know if this is mitochondria, what you do is overlay with the mitotracker, and the overlay was very good. And we further checked that the compound was in mitochondria by isolating the mitochondria. We were able to isolate mitochondria from cells and check if we have Reuterium there in the mitochondria. And you can check that by ICP-MS. And indeed, by using our compound, we observed that indeed we have a large amount of Reuterium that is in the mitochondria. However, if you use compounds that are not targeting mitochondria, for instance, these ones, or this one here, that goes to the cytosol, the amount of Reuterium in the mitochondria is much lower. So with our compound that we call p-pyru, we have a Reuterium complex that is mainly accumulated in the mitochondria of living cell. So the next question was, okay, we have the Reuterium in mitochondria, but is catalytic, can promote a chemical reaction? So to test that, we decided to go to the same reaction that Eric Megget had assayed, that is the removal of the alloc protecting group from rhodamine. And we were happy to see that our catalyst was able to generate green color in the cells. Also, the compound, the Reuterium that is not in the mitochondria, that is in the cytosol, also generate green color. You could see, well, this is similar, but it's not similar, because in this case, the overlay with the mitotracker was perfect, and in this case, there is a lot of green color that is not in the mitochondria. So by having the Reuterium in the mitochondria, we are generating the product, mainly in the mitochondria. So the next question was, and this is useful for something? Can this have an advantage over other type of biological tools? So for that, we made the following experiment. This is mitochondria to work, they need to have a membrane potential, because this is critical for the generation of ATP. And you can label the membrane potential by using this dye, this rhodamine derivative. If you destroy the membrane potential, then the color disappears. And for instance, if you treat cells with this compound, 2,4-dinitrophenol, the color disappears. Why? Because this compound destroys the mitochondrial potential. We need to use a lot. 500 micromolar is a lot. Why? Because this compound doesn't diffuses very well, doesn't go inside the cell very well, okay? But it works. But however, if you just introduce an allele group here, no OH, just allele, the compound is inactive, inert. So this is active, this is inactive. This could be a prodrug for this compound. And then we made the following experiment. We prepare two different sets of cells. We add our ruthenium to one of them, so this ruthenium will go to the mitochondria, and this one will go to the cytosol. Now we added the inert compound, and with this compound that is in the cytosol, nothing happened. However, we see this other compound that is in the mitochondria, we observed a very fast depolarization of the mitochondria. So what is going on here? What we think is happening here is that this compound diffuses very well through the cell, and when it reaches the mitochondria, it meets ruthenium, and then undergoes an explosive transformation into the active compound. So by generating the active compound in situ, in the organelle in which it has to work, we have a much more efficient biological effect. Maybe in the future, I don't know, this type of tactic can have therapeutical applications. So generating the active compound specifically in the cell organelle. So after this discovery, we made more experiments. For instance, we demonstrated that we are not limited to alloc protecting groups, but to allele. This is more robust. This is much better. That is more robust. And then we can do the same type of deprotection. And this is quite nice because we can even make a real-time movie to see metal catalyzed transformation in a cell. So these are two different sets of cells, left and right. I'm going to add the ruthenium only to this set of cells, not to the other ones. And then I add the precursors. So what should happen here? Well, I should have reaction only here because it's where I have the catalyst, not here that I don't have the catalyst. And this is what happened. This is a real-time movie, so we were recording what is happening in the microscope. As you can see, the green color of the product is only generated in the cells that contain the ruthenium catalyst. This is a demonstration that we have a ruthenium catalyzed transformation inside a mammalian cell. So ruthenium, why ruthenium? What about other metals? Well, we try other metals, and we liked gold, because part of our work in catalysis was in gold catalysis. Can gold catalysts also promote these type of transformations? Well, we first designed a probe that could be okay for the experiments, because this is not fluorescent, but after the gold catalysis, we obtained this coumarin derivative that is fluorescent, and we tested this transformation. I mean, this transformation has a problem, is that the gold, to be catalytically active, you need to remove the chloride. And this is the reason why we add the silver salt. So this is a problem, because we cannot add silver salts to cells. So this doesn't work if you don't have the silver salts. But Christian, who was a postdoctoral student, a brilliant postdoctoral student in my group, came to my office and said, well, you know, we don't need the silver salt. We just need to add excess of water. Water? Yes, excess of water. But this is ideal, because, you know, we want to do the reaction in water, and water is activating the catalyst. Yes, water is activating the catalyst because water is solvating the chloride and helping in the removing of the gold chloride. So we can do the reaction in the presence of water without the silver salt. So we were in conditions to test if the reaction could work inside a living mammalian cell. And we were happy to see that this works very well. If you have the precursor, it doesn't work. If you have the gold and then the probe, then the reaction works. And then with these results, we made one of the experiments that I like more in my brief history in this field, is playing with two different chemical reactions at the same time. So this is the probe for the gold, and this is the probe for the ruthenium. Both are not fluorescent. So if you add both probes, you see no color, no fluorescence, okay? So what we are going to do now is to add ruthenium. If we add ruthenium, then we remove this protecting group, and we get this product that is red, and it is what happens. If we add ruthenium, we obtain cells labeled with ruthenium. What happens if we add gold? If we add gold, we obtain the product of the gold catalysis. If we add both, we obtain both colors. So what do we have here? Well, we have two abiotic artificial substrates. We have two artificial catalysts that are able to work inside a mammalian cell that is alive and create an artificial metabolism that is compatible with the metabolism, with the natural metabolism of the cell. This is a field that is starting to emerge, this combination of artificial with natural metabolism, because there are many options. For instance, you could combine artificial metal catalysts with natural enzymes and then start to make hybrid metabolisms. So I'm quite excited about this research. I think this is something that many groups now in the world are starting to work on that, but we were one of the first to publish this type of artificial, very primitive metabolism that is non-natural and can be complementary to the cells. I'm not sure, Eugenio, how much time do I have? You were a lot of time in the table, you know, so. 10, 15 minutes? 15 minutes, okay. Okay, so I will, 10, 15 minutes will be okay, okay. So, as I showed you at the beginning, we were always very interested in cycloaddition chemistry, in metal catalysis cycloaddition chemistry, and curiously, one of the more important reactions in bioorthogonal chemistry is a cycloaddition. You know it very well, Nazario. It's the famous click, the copper catalyzed alkyne acid cycloaddition. No? It's the mother of bioorthogonal chemistry. And this is a metal catalyzed reaction, so the metal, the copper, is just doing what I told you at the beginning. It's just activating the substrates, the acid and the alkyne, and putting them together so that the reaction can work. But this reaction has a problem. If you want to do the reaction really in a living environment, the copper is very toxic and also very labile. The copper one tends to change the redox state, so it's not a good reaction to work in living cells. And so many people that was trying to apply this chemistry was in the membrane, in the outside of the cell, okay? Caroline Bertocci, for instance. Well, we were working trying to develop a ruthenium version of the click, and indeed we had an interesting couple of contributions using this ruthenium catalyst. We call it ruthenium acid thioalkyne cycloaddition because we use thioalkynes, as you can see here. And yeah, the reaction worked quite well, but you know, we were not able to translate this chemistry to cells. Why? Well, for several reasons. For instance, one of the reasons is that we have three components, 1, 2, and 3, the metal, the alkyne, and the acid. And probabilistically, the possibilities that they meet together in the environment of a cell is very low. And the other thing is that once you make this type of complex, probably the equilibrium in the cellular medium will go to the free alkyne. So we thought, maybe we can solve that. How? If we convert this alkyne into a bis alkyne. Because with the bis alkyne, it's like my two legs. It's like a twister. I will fish the metal in the soup of the cell. And we thought that this could be a good idea. And additionally, we know that once you have this type of complexes, you can make these metallocycles that can be intersected with alkynes and give this complex cycloaddition 2 + 2 + 2 cycloaddition to give everything. So I asked Joan to test if the reaction could be possible to carry out in a complex medium like a live cell. He first tested the reaction in cell lysates that are just dead cells in some way. And the reaction worked. The yield was not very good, but for us was interesting. We tested with this probe because the product is fluorescent, and we went to the cells, and this is what happened. So if you add these two substrates, you see nothing. If you add the product, what do you see? Well, you see that the compound is in cells, but you have a lot of diffuse color because there are a lot of compound in the membranes. However, if you generate the compound inside the cell, look, it is very clean. So it's another demonstration of the power of this technology, that if you make the compounds in situ, you can really produce this type of products that otherwise have difficult to enter into the cell. And this was even more surprising with this product. This product is an aggregation-induced emission dye because it contains this unit, tetraphenyl ethylene, that is aggregates. And because it aggregates, it doesn't go inside. Look, this is a cell. Look at the product. The product is outside. It's aggregating outside the cell. We cannot go inside. So what happens if we make it inside the cell? Well, if we make it inside the cell by doing our reaction, then the compound, the product is in the cell. And this was a fantastic demonstration, another possible application of this technology to generate products that otherwise are difficult to go inside the cell. So we made this type of reaction. We discovered others that I don't have time to talk about, but this is the idea that indeed, at the beginning, we thought that it would be not possible to achieve organometallic chemistry inside cells, but it's possible. Of course, the efficiency is very low, and we need, there is a lot of space to improve. I want to comment to you also another reaction that we have been able to achieve inside cells, but is not mediated by organometallic catalysis. This is a collaboration with Pablo Del Pino, that is a group working in nanotechnology and also in MOFs, in the CIQUS. And I realized that Pablo was making this type of interesting MOFs containing a gold nanostar inside the MOF. The MOF that they use is the famous CIF8. These MOFs tend to decompose in water, but they found that if you use this polymeric system, then the MOF with the nanostar is more or less stable in water. And even they were able to find that if you introduce a dye in the MOF, the MOF survives inside the cell, in vesicles inside the cell. So, and we knew, and I was talking to him, you know, with this gold nanostar, we can use the plasmonic properties of the nanostar to generate heat, these photothermal plasmonic properties. And this could be a good idea to generate heat. And can we promote reactions just by heating? I mean, you know, the ideal reaction could be one, but we prefer to test another one because it was easier to design a probe that could give something fluorescent. And we tested this one, that is nucleophile substitution. And this is if you heat for 1 hour, you get this product and fluorescent coumarin. But if you heat 5 minutes, the yield is very low. However, Pablo found that by using near-infrared light and the MOF, then you get almost 40% yield. And so it was working, probably because the compound was absorbed in the MOF, and then the heating, the local heating was very efficient. The good thing is that we were able to do that also inside cells, and otherwise you cannot heat cells at 80 degrees, but however, by using this system, you can promote thermal reactions inside cells. So it's a quite interesting and nice collaboration. So, and just to finish, coming back to the Metalloenzymes, so where are we now? Well, we demonstrate that a small metal catalyst can promote reactions in cells. Advantages and disadvantages with respect to Metalloenzymes. Advantage is the scope is much better than natural Metalloenzymes. I can use metals that natural Metalloenzymes don't have. The compounds are small and they can diffuse inside the cell. Disadvantages, low turnover, low rates, deactivation, they are rapidly deactivated. So what are we trying to make now? A compromise. So we are trying to make something that contains the metal, but also contains some part of the enzyme so that they could be more stable. And then there is a field that is growing, the field of artificial Metalloenzymes. But most of the people are trying to do that by using molecular biology. In our case, we are using our synthetic chemistry. And I will show you just one example. And with that, I will finish, of one of the initial mini Metalloenzymes that we are doing. And this is also come back to my work in the area of DNA binding. These are natural proteins that bind DNA to specific DNA sites. This is the natural protein, and this is a synthetic dimeric mini protein that also binds the same site. Currently, you have only the monomer. The interaction is very weak. It makes sense because you have less non-covalent interactions. But we were curious to know if we could improve the binding of this just only one monomer of the peptide. It's important to say that this monomer is unfolded in the absence of DNA. So the alpha helical folding is only in the presence of DNA. And we thought, okay, if we facilitate the folding, maybe we decrease the entropic cost of the association, and then we can promote the binding of the monomer. And this is what we did. So the peptide by itself is not able to do that. However, we thought that this could be possible by using a metal, because the metal will facilitate the alpha helical folding, and then we could have the interaction. And this is what Soraya and Natalia and Jessica found, that if you add this palladium complex, we promote the alpha helical folding, and we have DNA binding. This was further tested by circular dichoresis. This is the typical circular dichoresis of an alpha helix. In the presence of palladium, you have the alpha helix. Without palladium, no alpha helix. And it was quite funny to see that we can control the DNA binding. So if we add palladium, we have binding. But if we add a chelating agent that extracts the palladium, you decrease the binding. You remove the binding. And you can do it in a cyclic way. DNA binding, DNA unbinding, binding, unbinding. So you can decide. You have a molecule that you can say, okay, I want you to bind DNA, I want you to leave DNA, and we can control that. No? But so this is the more interesting thing that Soraya also discovered is that this peptide, in the absence of DNA by itself, is not able to go inside the cells. So this is just a color and a dye. See, very small amount. But adding the palladium, look, the compound is inside. So the palladium complex is helping in the internalization of the peptide. Why? Because the palladium is promoting the alpha helical folding, and when the peptide is alpha helical, it goes much better inside the cell. So the palladium is promoting the cellular internalization, but on the other way, this can also be observed as the peptide is promoting the internalization of palladium. No? And palladium is a very powerful catalyst. It's very difficult to use palladium in this complex medium because it gets rapidly deactivated. But we thought that by complexing the palladium with the peptide, we could really perform catalysis in cells with this type of complexes, and we demonstrate that this is possible, not with this catalyst, because these palladium are not working because these ligands are very strong. There is no space for the probe to bind to the palladium coordination sphere. However, if we use another palladium with a dichloride in this position, we can promote the intracellular transformation. So with more time, more transformation, we can use different probes. And look, this is a very important point. Our palladium peptide complex is able to promote the catalytic transformation. This reaction is the one that we promote. However, if you use the palladium complex by itself, the reaction doesn't work. So the peptide, the mini protein, is really helping in keeping the palladium active for the protection. So what we have here is a palladium peptide complex that we could call a non-natural mini metalloenzyme that is able to work in the same environment in which enzymes work. And we are further working in this type of projects. So I hope I have shown you that indeed this type of metals that are very powerful in human-invented organometallic chemistry can also be transferred to the field of cellular biology. So all this work has been possible thanks to the more important asset that we have, that are people. This is a quite pre-pandemic picture in the Ribeira Sacra. Many of you can identify. Indeed, half of the group has been already changed. But people are really the important thing in all this stuff. We also found some time to do some export before the pandemic. Somebody criticized this picture because there's only one girl. I have more now. But yeah, it was very, very nice. You are going to discuss later about talent attraction. Without talent, you cannot do anything. So when I started CIQUS, I said, I just want two things. I want the best PIs and I want the best students. This is easy. And if you have the best, then everything will come out. I like this. I'm not sure you know Daniel Thachman, but he was the president of the Weizmann for several years, and he used to say that the more important thing for science are not microscopes, but the eyes and the brain of people that are looking at the microscope. And also I like this other one, when Weizmann was asked about which field will be the one for the next scientific revolution, and he answered, I don't know which will be the field, but what I know is that the next scientific revolution will be driven by scientists that have a multidisciplinary view of science, the opportunity to take risks, the infrastructure to work, and the freedom to think. The freedom to think, I like that. And I will add, and the time to think. That is something very, very important. I'm sure you do the same. I always ask the politicians and the members of the university, whatever, what we need is time. This is the more important and precious jewel for us. So time and freedom. So this is possible. CIQUS, this is a picture of CIQUS. We are over 170 people in total. And this is in Santiago, in Galicia. You know, sometimes working in Galicia is not easy, and Maria is around. She probably agrees with me. In part because you can find things like this. You know, you are driving and then you go to, this is in my land in Orense, and so which is the pathway that should take, you know? This is Galicia. So to finish, I want to announce that we are younger than ICMol, but we are celebrating the 10th anniversary this year. Hopefully it will be possible to celebrate it, and it will be at the end of March in Santiago. We were lucky because I had talked to Dave McMillan before he got the Nobel Prize, so probably the first trip to Europe will be to Santiago for David McMillan. And so we have assembled a very nice panel of speakers. Maria Jimenez, that is there, has been involved in the organization also. So we are invited to come to Santiago at the end of March if the COVID situation is allowing us. So I think I talked too much, so I stop here. Thank you.
Well, thank you very much, José Luis, for this fantastic talk, and time is open. The floor is open for questions. Okay, let's start with Nazario.
Thank you so much for this wonderful lecture, disruptive, highly disruptive chemistry, and therefore many things to do. First, I like to start with a comment, because I guess that the most important you are facing is, first of all, internationalization. Internationalization.
Internationalization also.
Yeah, also is also important. Yeah, and selectivity, because these metals are very active, as you mentioned, and therefore my first question is related what is going on in other organelles in addition to mitochondria and so on.
Yeah.
And just to finish with my question is, in the second part of your Diels-Alder cycloaddition, particularly the related with the click chemistry reaction, mostly click chemistry reaction, when you remove the catalyst, the copper, I was expecting the strain-promoted cycloaddition with the cyclooctane instead of ruthenium. But again, it is metal is critical, but many other reactions like this could be also performed.
Okay. Okay, with respect to the first question, what we usually do is to follow the toxicity of our systems completely. We study different forms of toxicity, membrane toxicity, cytotoxicity, antiproliferative activities. So this is what we can monitor. You know, we also monitor by ICP where is our metal, in which organs we can do that. And this is what we usually do as tests. But we cannot really make a detailed track of the fate and also the damages that our metals can really promote at the molecular level. But you know, with ruthenium, it's surprising. Cells survive for 24 hours like if you were adding nothing. So ruthenium is a fantastic biocompatible metal with different type of ligands. So gold is more complex, palladium is difficult, but ruthenium is the one that is giving the best results. So we are quite confident that if we are able to increase the turnover with some of our ruthenium catalysts, perhaps by involving them in MOFs, for instance, or in proteins, in mini proteins, we can get closer to enzymes, but with the advantage of a better scope and with the advantage of using a transition metal, so a reaction that is not known for nature. With respect to the click chemistry, we use thioalkynes. And the use of the thiol is critical for the ruthenium catalyzed reaction. If you don't have the ruthenium, the reaction doesn't work, of course. And the mechanism, we have studied the mechanism, we know the mechanism. And the advantage of, I mean, when you want to do a click ligation, in some cases it's good not to have a metal. I understand that. But this is also dangerous because you have to use strained reagents. And these strained reagents in complex media can undergo reactions, for instance, addition of thiols, glutathione, for instance. And then this can be dangerous. But in other cases, the metal gives you another advantage, is that imagine you have two different types of cells, and you have metal only in one of them. Or you can drive a metal to a specific type of cell, to cancer cells. You could do a click reaction only in cancer cells because it's driven by the metal. So the metal has advantages and disadvantages. But of course, I want to sell the advantages.
Thank you so much. Maybe Macmillan can give you some ideas.
Well, Macmillan, you know, we were starting to do this photocatalysis for all these transformations over 5 years ago. But things go slow in our labs, and now Macmillan and other people are doing that. So they kill us in some way. But it's difficult to compete sometimes.
Yeah, it's always wonderful to have you here again,
Oh, hello.
José Luis. So I just have two questions, or one question and one comment is, even if you don't see any cytotoxicity in the cells, do you see any ROS, reactive oxygen generation of this metal complex inside the cells? Because there are metals they can generate, obviously is something that take into account. And the second is, do you think about using, because I mean your final output is always the microscopic output of the fluorescence, to use any nucleotide type derivative and use sequencing methods to visualize the incorporation of this into DNA of the mitochondria or similar?
Yeah, okay. So the first question, we don't analyze all the days if they are producing ROS, but we have the methods, and in some cases when we have good results, this is another test that we used to do to see whether there is a ROS increase, but this type of ruthenium complexes that we use are not particularly good for ROS, okay, so there is no problem, and in most of the cases to generate ROS you need photochemical activation. And the second question is, yes, I use microscopy because it's the easier ways to follow, but I can tell you now that we have a methodology already working that is based on HPLC mass spectrometry detection of the products. So we can follow the reactions by using LC mass spectrometry.
Thank you. Any further questions?
So thank you very much for your nice talk. It was really, really interesting, but also like dreaming somehow. I mean, it's giving a lot of ideas for the future. I was a little bit afraid that Jorge and Nazario would have spoiled one of my questions, and you actually spoiled one of my questions by replying to them. I was really willing to ask about cytotoxicity. I have another question. You show about the MOF complex with ruthenium, and you show also about the near-infrared irradiation. And the temperature, it looks like arriving until 80 degrees, even in cells. So normally 80 degrees in cells, I'm expecting the die of the cell, the death of the cells. So what about it? This is my first question. And a second question, but this is really like a really future perspective. What do you think about the formation through this, maybe through this kind of catalysis of nanoparticles inside the cells? I mean, synthesis of nanoparticles inside the cells that then can be used, in example, spoiled, exploited to generate ROS or generate heat and kill cells. Thank you.
Yeah, the second question is something very interesting. We are also trying to develop supramolecular assemblies promoted by metals inside the cell. This is a collaboration with Juan, for instance. So anything that you can really synthesize inside the cell in a controlled and human-invented way, I think is great, you know, because we don't have the time that nature has. Nature has all the time for evolve, but we only have a few years to work. So we need to overcome nature, you know, in some way. So I like human-invented things. So it's a good idea. And the first question was no. Maybe I didn't show because it was very fast. No. If you do the reaction by heating, the reaction without MOFs, by heating, you need over 80 degrees and 1 hour. Okay. But if you use the MOFs, the nice thing is that you can promote the transformation with the localized heating that is generated inside the MOF. Yeah, with the lasers, you irradiate the MOFs, and then there is a localized heat. And interesting, the MOF is also is isolating the how do you call this? The thermolysis. The heat is not expanding, you know, and then you have the concentrated heat in the MOF, in the pores, and then you don't. The bath is cold, so the heating is just local. Okay, so.
Okay, well, I have the last question.
Okay.
I'm wondering about the toxicity of your treatment with the cells. I don't know, you have just tried to record the life of the cells after the treatment, after the reaction. And also the reversibility, if you can use the cells as a catalyst, for example.
Yeah, this is very interesting. This is very interesting because we have been able to, the toxicity, yes, we have the controls because sometimes we observe toxicity, but other times we don't observe toxicity. So, but this is a control that we need to do all the time, the toxicity. We have different ways of controlling toxicity. So we do that. But developing catalytic cells, I like that, you know. And can you make a catalytic tissue, so and implant it in a mice? I like that. This is something that we were talking about also. Thinking.
Well, I think we can go for the coffee. Join me to thank the speaker again for his wonderful talk. And